Hey, I'm Junior from UCLA. In this video, I'm going to introduce our work, JEDI, CircRNA prediction based on junction encoders and deep interaction among splice sites. This is a joint work with Chelsea, Junhan, Muhao, and Wei. Starting from the introduction, CircRNA is a novel class of long non-coding RNAs with non-canonical splicing processes called backsplicing. This figure shows the difference between backsplicing and canonical splicing. Backsplicing will result in closed continuous loop, where the 5' end splice site is ligated to the 3' end of the downstream site. There are also several benefits of circular structure, such as RNA amplification, genetic information rearrangement, and RNA folding constraint. The task of the CircRNA prediction is very important because there are more and more emerging evidences that suggest critical roles of CircRNA, such as sponges for microRNA, protein interactome, host gene transcription induction, and RNA binding protein competition. As a result, high-throughput CircRNA prediction becomes an essential step for exploration. In the previous studies, there are two lines of research about CircRNA prediction. The first one is about expression-based methods. They search for some chimeric risks they join from 3' end sites to the upstream 5' end site. However, there are several caveats. They are very sensitive to expression abundance, but CircRNAs are often lowly expressed with a low sequencing coverage. Some other drawbacks included long duration, high range usage, and complicated pipeline. The other line of research is about sequence-based method. Some methods extract discriminative features and apply some conventional machine learning models, but those features are only for some specific and limited facets of a sequence properties. Some other methods apply deep learning techniques, but many of them just ignore the important positional information on sequences. They also have very limited coverage on interaction between donors and acceptors. To address these challenges, in this work, we propose a novel framework, JEDI, Junction Encoder with Deep Interaction. This is an end-to-end deep learning framework to predict the CircRNA based on the sequence. For all of the junctions of the acceptors and the donors, we use the junction encoder to derive the acceptor and donor embeddings. To mimic the backsplicing behaviors, we propose a novel cross-attention by observing the interaction between donors and acceptors and derive the cross-attentive acceptor and donor embeddings. To have the final features, we further use the acceptor and donor attention to divide the final acceptor and donor representations and use the fully connected layer to have the final prediction. For the attentive junction encoders, we're starting from learning the k-mer embeddings that can preserve a complicated local context. Based on the k-mer embeddings, we apply the bidirectional recurrent neural nets to learn sequential properties in the genes. Finally, the k-mer attention is used to estimate the unequal importance over different positions in the genes and have a weighty aggregation of their representations. To implicitly learn the backsplicing behaviors, we propose the cross-attention for modeling deep interactions between acceptors and donors. The cross-attention weight between an acceptor and a donor can be estimated by the dot product similarity between their embeddings and the softmax function. Based on the cross-attention weights, we can estimate the cross-attentive acceptor embeddings and the donor embeddings based on the weighty combination. We further propose the attention-based feature extraction method for both acceptors and donors. The acceptor attention scores and the donor attention score can be estimated by a dot product similarity to a learnable attention vector for either acceptor and donor. and we still use the softmax function to have the normalization. Finally, we can have the ultimate representations for both acceptor and donors by the weighted aggregation. Treating them as the features, we can use a fully connected layer to have the final prediction and treat the whole problem as a binary classification. For machine learning and optimization, we solve the task of the CircRNA prediction as a binary classification problem. We optimize our JEDI model with the binary cross entropy as the loss function and use the L2 regularization to avoid some overfitting phenomenon. We also want to mention the interpretability of our proposed model JEDI. Specifically, those attention scores are very properly maintained like a probabilistic distribution. In other words, we can treat it as some probability estimation. For example, the donor attention score for the donor J for prediction of CircRNA can be implicitly estimating the probability of starting a backsplicing from donor J. As a result, the variable attention score can be referred to the probability of starting a backsplicing from donor. Similarly, the cross attention score between donor J and the acceptor I can implicitly learn the conditional backsplicing behaviors from the donor J to the acceptor I. As a result, we can refer this variable to a conditional probability given the donor J to have the backsplicing event to the acceptor I. Based on these two observations, we can estimate the joint probability of having the backsplicing between the donor J and the acceptor I based on the multiplication of the two probability. So this joint probability can be divided into the multiplication of this marginal probability times this conditional probability. In our model, then we can simply use the multiplication of two variables to represent this probability. For experiments, we conduct experiments with two datasets. The first dataset is the human CircRNA dataset. This is the benchmark dataset generated by the previous work, including 32,000 isoforms of the human CircRNA covering diverse tissues and circular types from the CircRNA database. For the negative examples, we have 20,000 experimentally validated and manually annotated negative transcripts from the GenCode version 19. We also use the duplicated genes database to cluster those isoforms into 23,000 clusters, and we divide those clusters into 5 parts to conduct a 5-fold cross-validation to prevent some information leak. For the independent study, we also adopt the dataset of the mouse CircRNAs. There are about 2,000 small CircRNA from the Circ base and 1,500 annotated negative cases from the GenCode version M1. We treat it as the test dataset for the independent study. We compare our model JEDI with 8 competitive baseline methods, including several traditional machine learning and deep learning methods, some deep learning CircRNA prediction method, and one conventional long non-coding RNA prediction method. We evaluated the performance with 7 conventional binary classing metrics, including accuracy, precision, sensitivity, specificity, F1 score, MCC, and AUC. The first task is about isoform-level CircRNA prediction. Given the gene sequence and the splicing information of an isoform, we predict if the RNA isoform is a CircRNA. As a result shown in this table, we can see our model JEDI has an excellent performance over all of the evaluation metrics and outperform all of the competitors in this task of the isoform-level CircRNA prediction. The second task in the experiment is about gene-level CircRNA prediction. Different from the first task, we are given both of the gene sequence and all of the exon intra-boundaries. And in this task, we are going to predict whether any of the junction pairs can backsplice to form a CircRNA. As the result shown in this table, we can see the performance still remain similar. And also, our model JEDI still beats all of the baseline methods in this task of gene-level CircRNA prediction. For independent study, we want to ask if our model can be applied on some other data of other species. In this task, we perform the prediction on the mouse CircRNA, but all of the model can be only trained on the human CircRNAs. As a result shown in this table, we can see because of the different species in the testing dataset, all of the model have a droppy performance over all of the matrix. However, our model JEDI still remain a acceptable performance over all of the matrix and outperform all of the baseline methods. As a result, we can see the JEDI can still precisely predict mouse CircRNA, although they are only trained with the human CircRNAs. As mentioned in the interpretability about the CircRNA and the JEDI model, we can perform the zero-shot backsplicing discovery based on the JEDI's interpretability, so that we can retrieve some backsplicing junction pairs without using any training backsplicing site annotations. More specifically, we can rent all of the possible site pairs between acceptor and donors based on the estimated joint probability that we mentioned before. As a result, we can treat it as a ranking problem and evaluate with some metrics like AUC. This figure shows the retrieval performance over all of the 5 folds in the evaluation. As a result, we achieve about 80% of the AUC score with a very small standard deviation. It means that although we have no any training backsplice site annotation, our model can still reach very effective and stable retrieval performance. This result also implies some learned biological insight of JEDI, because we can learn some relations between the acceptors and donor without any annotation, based on only the information about the existence of the backsplicing. Finally, I will give some of the conclusions. First, JEDI is an end-to-end deep learning framework for the task of the CircRNA prediction. We first used the Attentive Junction encoder with a chemo embedding to derive the satisfactory representations. We proposed novel cross-attention layers to model the backsplicing behaviors and the interactions between the acceptors and donors. And the acceptor and donor attention finally performs a very well job for the robust feature extraction. In the experiment, the JEDI reaches a very effective performance on all of the tasks, including isoform-level, gene-level, and independent CircRNA prediction on both human and mouse datasets. Compared to the previous models, JEDI outperformed all of the state-of-the-art CircRNA prediction and conventional long non-coding RNA classification methods. We also demonstrate a strong interpretability of JEDI that enables the zero-shot backsplicing discovery and retrieve some of the potential backsplicing sites for validation. Finally, I want to thank all of my co-authors in this project, And for some other information, please check our paper in the code repository online. Thank you very much. If you have any question, please ask in the live Q&A session.